Okay, so welcome to this next video in the playlist on cyclic AMP signaling. In this video, what we're going to talk about is lipid rafts, and we're going to talk about lipid rafts so that we can talk about microdomains. So this video is going to be on lipid rafts and microdomains. Okay, and really these are, I don't know, more grown-up concepts in biology, basically. So you may have once viewed the cell, or you may even still view a cell, as just being a blob with a cell membrane around the outside, and then a huge mass of cytoplasm inside. Okay, well, you need to, these concepts are more grown up than that, basically. The cell isn't that simple. Instead, it's compartmentalized into loads of little functional compartments. And these little functional compartments, so let's say this is a little functional compartment which is sort of separated from the rest of the cell. Something is going to go on in this little compartment so that you can, you can keep all the stuff that's going on in this little compartment separate from the rest of the cell. And this is really an important concept for cyclic AMP signaling, because we need to get specificity of cyclic AMP signals. And if we just have cyclic AMP going up in the entire cytoplasm, that would cause a huge number of downstream pathways to activate. But you don't want that usually. You just want a specific response to be induced in this cell, one of the responses that cyclic AMP induces. And basically the way of achieving that is to have cyclic AMP going up just in one of these little microdomains, and then have all the effectors that are relevant also in this microdomain, and those will respond to that rise in cyclic AMP. But all the effectors of cyclic AMP that you don't want to be activated, you'll have those outside of the microdomain, and therefore they're safe from being activated. So it's a really important concept for how you achieve cyclic AMP specificity. So this is the concept of a microdomain, a small compartment of a cell in which certain signaling processes are going on. Okay, and a very important concept related to microdomains is the concept of a lipid raft. So we're firstly going to discuss the concept of a lipid raft, and then we'll see how lipid rafts are incorporated into the concept of a microdomain, along with other concepts that are important for microdomains. Right, okay, so lipid rafts. What is a lipid raft? And again, this is a grown-up concept, because you may have once viewed the cell membrane as being just a homogeneous layer of phospholipids, so being basically just having phospholipid after phospholipid after phospholipid, i.e. if you go to any portion of the cell membrane, the consistency of the phospholipid bilayer will be the same. That was the way that you may think the cell membrane is, but basically it's not true. The cell membrane is not homogeneous, so it is not homogeneous. So it is not like this picture here. Well, bits of it will be like that, but other bits of it will be very, very different. And floating in the phospholipid bilayer, what you have are these things called lipid rafts, and basically they are portions of the membrane which have a very different consistency to the usual phospholipid bilayer. So these are known as lipid rafts, and basically they're made up of loads of different types of lipids to the usual phospholipids. So these are phospholipids, which means a diacylglyceride molecule with a phosphate group bound to the third hydroxyl group of the glycerol. So let me just show you the structure of one of these phospholipids. So the usual structure of one of these phospholipids is that the whole thing is based on glycerol. Glycerol is propane 1,2,3 triol, so it has 3 hydroxyl groups coming off, a hydroxyl group here and a hydroxyl group here. In the phospholipid, they're all going to be bound to stuff, so that's why I haven't drawn the hydrogens on. But you will have hydrogens saturating these carbons, like so. Right, now, to make a phospholipid, what you do is you ester link a free fatty acid, basically, to each of these 2 hydroxyl groups. So we have 2 long-chain carboxylic acids, or free fatty acids, so I'll highlight those as free fatty acids. So these are long-chain carboxylic acids, or more commonly known as free fatty acids. So long-chain carboxylic acids, which have been ester linked to the hydroxyl groups on two of these hydroxyl groups on the glycerol molecule. Long chain carboxylic acids / free fatty acids. Well, they're not free anymore, so I'll call them fatty acids. Right, okay, and then we have our glycerol molecule, and the third hydroxyl group of our glycerol molecule is basically bound to a phosphate group, so I'll put in a phosphate group here. Okay, so there's our phosphate group, and you can see that this phosphate group is polar. It's got a negative charge here, whereas these have absolutely no charge on them. They are very unpolar. This likes to interact with water because water is a polar molecule as well. It has a positive. Water has a positive side and a negative side. So here is water, basically. Because oxygen has a greater electronegativity than hydrogen, the electrons in these two bonds are more attracted to the nucleus of oxygen than they are to the nucleus of hydrogen. So basically they spend more time around the oxygen than they do around the hydrogen. So overall get a partial negative charge on the oxygen and partial positive charges on the hydrogens. So these partial positive charges can interact with this polar group here. And that's why phospholipids, when we draw this structure like this, this little dot here, this head, refers to this phosphate group here. And these two tails here clearly refer to those free fatty acids back there. So the polar head, this phosphate group, faces out into either the extracellular fluid here, so this is the extracellular fluid, or it faces in towards the cytoplasm. And that's because the cytoplasm and extracellular fluid have water in them, so that's why the polar heads face outwards, so that they can interact with water. The hydrophobic tails, which can't really interact with water, or it's very unenergetically favourable for them to interact with water, those face in towards one another, and they sort of like being amongst each other because they're very lipophilic, so they like other lipids. So that's why this structure makes chemical sense. Okay, so that's what usually this membrane is made up of. These lipid rafts are basically huge collections of the membrane where you do not have this structure, basically. Instead, you make the membrane out of different types of lipid molecules. So, basically they are made out of different type of lipid molecules. Now, one of the type of lipid molecules you use is what are known as sphingolipids rather than phospholipids. So, let me show you what the structure of sphingolipid is. Now, sphingolipids are all based on the structure of a molecule known as sphingosine. So, firstly, what we need to do is look at the structure of sphingosine. So, sphingosine is an 18-carbon molecule, so I'll draw out the full structure of sphingosine. So 4, 5, 6, 7, 8, 9, we're going to run out of space, 10, 11, 12, we're going to have to get smaller, 13, 14, 15, 16, 17, 18. I think that is 18 carbons. Right, so sphingosine, you have a hydroxyl group on this first carbon, then an amino group on the second carbon, another hydroxyl group on the third carbon, and then a double bond between this fourth carbon and this fifth carbon. That's all the structures you have to remember in sphingosine. Then just put hydrogens on everywhere else to saturate it, and then you've got the structure of sphingosine. So this is the structure of sphingosine. And sphingosine is the basis for sphingolipids. So I'll show you how to turn this molecule into what is known as a sphingolipid. So firstly, before we can turn it into a sphingolipid, what we need to discuss is how to turn it into a molecule known as a ceramide, because ceramides come before sphingolipids. You turn sphingosine into a ceramide, and then you can turn a ceramide into a sphingolipid. Okay, so I've almost finished saturating my sphingosine molecule now. All these little hydrogens here, and then finally there. So I've finished the picture now. So that's the structure of sphingosine. Right, so to turn this into a ceramide, what you do is, again, you take a long chain carboxylic acid or a free fatty acid. So here is our free fatty acid, and it will have some R group here, and I probably should put R prime since I've already used R and R prime. Now, Basically, the way you turn this molecule into a ceramide is by binding a free fatty acid to this amino group here to create an amide group. So basically, if I draw out this amino group in full, so I take off those two hydrogens and draw them properly, so the nitrogen bonded to two hydrogens, basically, what you're going to do is you're going to take off a hydrogen from the amino group. You're going to take off the hydroxyl from the carboxylic acid, and then you're going to bind that hydroxyl group to the hydrogen to make water, and you're then going to bind the nitrogen to the carbon, like so. And that's going to create you an amide link there, okay? So that is what is known as a ceramide. Sphingosine linked by an amide link, hence this amide in the ceramide name, to a long-chain carboxylic acid. And this R prime group here can be whatever you want it to be. It can be whatever aliphatic chain you like, basically. Whatever it is, it's still a ceramide. All of the, whatever you can put there, all of those molecules are ceramides, basically. Now, to turn a ceramide into a sphingolipid, and in fact, all ceramides actually are sphingolipids, but ceramides are like kind of trivial sphingolipids. What you do is you bind something to this hydroxyl group up here. So, in the case of ceramides, what you have done is you have bound hydrogen to the hydroxyl, to the oxygen, basically. So ceramides are sphingolipids. It's just where the group bound to this oxygen is a hydrogen. But more generally, you're going to replace that hydrogen with an R group, and that is a sphingolipid. Anything with this structure where you have sphingosine with an R group here, any old group here, and a carboxylic acid bound to this amine group of sphingosine to make it a ceramide, basically. So all ceramides are sphingolipids, just where the R group here is a hydrogen, and basically all sphingolipids have two places where they can be variable. They have this group where they can be variable, and they have this group down here where they can be variable. Right, so that's the structure of a sphingolipid. Sphingolipid. Right, so now I just want to, now we're finished. Lipid rafts contain lots of these molecules, basically, in their membranes. So instead of having normal phospholipids, they have these sphingolipids. Now, they also have a specific type of sphingolipid is often very, very highly present in lipid rafts, and those are known as gangliosides. Specifically, there is a very special ganglioside called the GM1 ganglioside, and that's very, very famous. The reason being, it's the ganglioside, the molecule that's in cell membranes to which cholera toxin binds. So that's why that ganglioside is famous. And in these gangliosides, this GM1 ganglioside is very highly present in lipid rafts. Now, what is a ganglioside? Well, gangliosides are sphingolipids, and they're a special type of sphingolipids. They are what are known as glycosphingolipids. And a glycosphingolipid is basically where this R group that you've attached to this this oxygen atom over here is a sugar. That's why you've got that glyco. Glyco means sugar from some old language. Basically you attach a sugar group here, some sort of large sugar group, some carbohydrate, and then your molecule is known as a glycosphingolipid. Gangliosides are a type of glycosphingolipid. You attach a really large sugar group on here and you get a ganglioside. And the GM1 ganglioside is a specific one of these gangliosides, and it also happens to be the one to which cholera toxin binds. So that's how cholera toxin gets into the cell, by binding to GM1 ganglioside. Okay, so that's a little aside. But these GM1 gangliosides are very highly present in lipid rafts. Now, so we've covered sphingolipids, we've covered gangliosides. There are two other really important molecules that are found in lipid rafts, which I can't really neglect, rather. They are cholesterol and ergosterol. So these are two other molecules that you will find in lipid rafts. So let's go over the structures of cholesterol and ergosterol. Right, so they both have this suffix. They both have in their names this suffix sterol. Now, the sterol molecule, the sterol structure rather, is based on the steroid structure. So before we can do the sterol structure, we need to look at what the steroid structure is. And basically the steroid structure, when anyone talks about a steroid, it's a chemical classification, not a biological classification, it's a chemical classification. And it means that the molecule's structure has one of these in it. So this is the steroid structure. So you have lots of carbon rings stuck together basically, like so. So you have two hexagons of carbon stuck to one another like this. And by the way, when you're drawing steroid structures, it's very, very common to use skeletal structures rather than using actual atom by atom structures like this. Just because, well, it would have been convenient to use a skeletal structure here so that I didn't have to draw all of these hydrogens on. So basically, just to remind you of what skeletal structure means, you don't draw carbons. Whenever there's a corner, that means a carbon. Implicitly, that means that you have a carbon there. And you don't draw hydrogens at all. So if you have a carbon here, you can see that there are two bonds off this carbon linking to other carbons. But what are its other bonds? Unless stated otherwise, it's assumed that the other bonds that are missing, basically, in this picture are just hydrogens. It's just bonded to hydrogens. If it's not bonded to hydrogen, then you draw it on. Okay, so basically, the steroid structure is like this, skeletally drawn. You have another hexagon ring up there, and then finally a pentagon ring, a 5-membered ring here. That is what the structure of a steroid is. If you have one of these structures or something very similar to that in you, in your molecular structure, then you are a steroid. Now, how do you turn yourself into a sterol? What's the difference between a steroid and a sterol? Well, what does the O bit usually mean? Alcohol, ethanol, methanol. It means usually hydroxyl group, an alcohol group, and indeed that's what you do have. You have a hydroxyl group there, and you are a sterol. So I'll highlight that in. That turns you into a sterol, putting that hydroxyl group there. Okay, right. So that's the general structure of a sterol. Now what we'll do is look at the specific structures of cholesterol and ergosterol. So I'll get another piece of paper. Right, so the structure of cholesterol we'll start off with first. So I draw out the sterol structure again, which is this 6-membered, well, these 4 rings, 3 of which are 6-membered and 1 of which is 5-membered. So here's the second 6-membered ring, the third 6-membered ring, and finally this 5-membered ring up here. Okay, right. I also put on the hydroxyl group because it's a sterol. And now there are some special groups that you add on to turn it into cholesterol. You put a methyl group off this carbon here. So now I've got a methyl group off there. So that this carbon here, this one here at this center, now has 4 bonds, all of them bonded to separate carbons. So it will have no hydrogens on. Okay, so that's a methyl group. This carbon has only 1 bond to another carbon, and it will have therefore 3 hydrogens off. You also put another methyl group up there. Okay, and then you put a double bond down there. So on skeletal structure, you show double bonds like that. And then you put on a structure here. So you put on a 6-carbon structure here. So that's 1 carbon, 2 carbons, 3 carbons, 4 carbons, 5 carbons, and 6 carbons. And then, to finally complete the cholesterol molecule, you stick off methyl groups up there and up there, which is quite a nice symmetric structure. I quite like that bit up there. It's nice and symmetric. Okay, so that is the structure of cholesterol. Okay, so you have lots of these molecules in lipid rafts. Now let's do the structure of ergosterol. Okay, so ergosterol has a very, very similar structure to cholesterol, so I will draw it actually on top of here. I'll draw the little modifications that you make to turn this molecule into ergosterol, and I'll draw them in pink. So, to turn this molecule into ergosterol, you firstly turn this bond here into a double bond. Secondly, you put another methyl group off down here. and finally you put another double bond there. Okay, and that turns you into Agosterol, basically. So in the pink we have Agosterol. Okay, so Ergosterol is basically just a modified form of cholesterol. So these two sterols are very highly present in lipid rafts. Okay, so we'll call it there for this video and continue on in the next video.